Okay, so these are the questions. These are the questions from energy and respiration, and that's May June 2023. We have already discussed this first question. We will briefly discuss the second question, and then we will start with the third question then. So this second question says that Oryza is a genus of grass plants that include the rice, Oryza sativa, a food crop. Farmers flood fields of rice because they encourages faster growth and higher yields. An adaptation of rice plants that allows them to grow in water is the development of parenchyma. State the function of parenchyma. So first of all, parenchyma is the special type of tissue in the rice stem which has hollow spaces in between. Within these hollow spaces, atmospheric oxygen can be and atmospheric air can be trapped. And what is the advantage and function of this parenchyma and this trapped oxygen? It supplies a continuous supply of oxygen, diffusion of gases can take place, and aerobic respiration can take place. Next question is saying state one other way in which the roots of rice are adapted to being submerged in water. So roots are adapted in multiple ways to being survive in to being in submerged in water. They can respire anaerobically to produce ethanol. The cells of the rice plant are tolerant to ethanol, so that is their adaptation, so that they can survive if only on anaerobic respiration even easily. Then excess of alcohol dehydrogenase is produced. It's an enzyme which converts ethanol back to ethanol. So this is how they can break the ethanol back to ethanol. Okay, back to ethanol. Just a minute. Okay, so and the last is I've told you that they have adventitious roots which allow the release of excess of ethanol and not excess, all the ethanol. And ethanol is a waste product, so it's wasted out in this way. Okay, next I have told you that these questions are from natural selection and artificial selection. That's not the topic. We are discussing the past paper questions of respiration, so we will not be discussing this. This is also from the chapter that is coordination auxin hormone, so we'll not be discussing this as well. And this part is the explain how these two species may have evolved through sympatric speciation. So that's from evolution and ecology and speciation. See, so this is a question which had the start of respiration but had multiple concepts in other parts of the question. So we'll not be discussing the questions related to not related to respiration. We'll be discussing only the questions related to respiration.
Okay, madam.
Okay, now this question that purely seems to be purely from an aerobic respiration. You can see glycolysis. It's showing glycolysis. You can see glycolysis is a biochemical pathway that occurs in the cytoplasm of cells. In glycolysis, a molecule of glucose is metabolized to two molecules of pyruvate. We know this is how this is happening, and at the end, two pyruvate are being formed. The process is outlined in figure 3.1. You can see this glucose and two ATPs are utilized. That's actually, as I've told you, is the investment phase in which two ATPs are being utilized. And this fructose 1,6-phosphate which is being formed is then converted into 2-triose phosphate, that is dihydroxyacetone. As I've told you, if you remember, dihydroxyacetone, and that is glyceraldehyde. And I told you that the reaction continues with glyceraldehyde, and this dihydroxyacetone is later on also converted into glyceraldehyde. Yeah, so this process is going to take place, and at once one AD is reduced and two ATPs are produced. And when this thing will repeat it again in the form of again that version of dihydroxyacetone and glyceraldehyde, so you know that total 2 Na DH are produced and total 4 ATP are produced, and when we subtract these 4 ATPs from these 2 ATPs which are being used in the first phase of glycolysis, so we get 2 ATPs in our hand in this way. So that these are the products of glycolysis: 2 NADH, 2 ATP and 2 pyruvate. So explain why glycolysis is phosphorylated at the beginning of glycolysis. See, I told you that this question is being asked almost every year, that why glucose is phosphorylated at the beginning. Why is it converted into glucose 6-phosphate, then fructose 6-phosphate, then fructose 1,6-biphosphate. Why phosphate is added to glucose? Remember, we discussed it. So why? Explain why glucose is phosphorylated at the beginning. Can you tell me?
उसको फ्रुक्टोज बिसफॉस्फेट में बनाता है, कन्वर्ट करता है.
Glucose is converted into glucose 6-phosphate. Na? So why phosphate is added to glucose? We have discussed it many times, Uruj.
उसको उसका शेप चेंज करने के लिए
Yeah,
मैम.
so that it is changed, it does not pass out of the cell through the same carrier
जी.
protein, and in this way it can not leave the cell. So glucose
ताकि वो ना
cannot,
जा सके, लीव करे.
yeah, it can't leave
शेप
the
चेंज
cell. Very good. Secondly, whenever phosphate is added to anything, if it's added to nucleotide, if it is added to ADP, when phosphate is added to ADP, it becomes ATP. It gets energetic. It gets
जी.
energy of phosphate. It gets energy because of the bond is formed between phosphate and ADP. So it also, addition of phosphate also activates. It also activates the glucose. Okay, glucose is made more reactive because of the addition of phosphate. Glucose becomes more reactive because of the addition of phosphate. Then it cannot leave the cell. Its shape is changed. Phosphate activates glucose. Its energy level is increased. All these can be the answers. You just had to write one. Energy level is increased. Okay, then the next question is, suggest one use of reduced NAD that is produced in glycolysis. Yes, what is the use of reduced NAD? Can you tell me that the reduced NAD this is produced, what does it do?
हाइड्रोजन को कैरी करते हैं.
Very good.
स्टेप
It's
से.
an excellent. Root, it's a hydrogen acceptor, it's electron acceptor.
जी.
It provides
जी.
hydrogen protons, and then at the end it is used for redox reaction, means it itself is oxidized at the end and it reduces the other things. So it is used for redox reaction, and simply it is an carrier of electron and proton, and it provides proton hydrogen ions to the electron transport chain. Provides hydrogen ions and electrons to the electron transport chain at the end. Okay. Now name the type of phosphorylation reaction by which ATP is made during glycolysis. Can you tell me which type of phosphorylation is this in which ATP is produced during glycolysis?
Substrate level
Excellent,
phosphorylation.
excellent, very good. That is substrate level, or you can call it substrate linked phosphorylation, because in this no oxygen is involved, in this no ATP synthase is involved. So when these things are not involved, it's substrate linked or substrate level phosphorylation, in which one substrate is just broken down, and from that substrate phosphate is added to the ADP to make it ATP. ATP synthase is not involved in it. Pyruvate can enter the mito. See, this is all these questions are from about glycolysis, and it's
जी
a long question of 9 marks, having short questions all related to glycolysis. So all the steps of glycolysis are very important. Pyruvate can enter the mitochondrion by active transport. We know it that it enters actively transported into the mitochondria. Describe
जी
the main conditions that are required for pyruvate to enter the mitochondria by active transport.
ये
Yes. What are the things needed? What are the things actually needed for active transport? That is what you just have to tell me.
हम्म,
Beta, first of all, active
ठीक
transport.
था
Uh-huh.
जी जी मैम बोले बोले मैम
It has to be actively transported. So first of all, oxygen should be available.
जी
ऑक्सीजन होगी तो ये जाता है ना, अदरवाइज तो इट द पायरुवेट इज शिफ्टेड टुवर्ड्स एनारोबिक रेस्पिरेशन, सो ऑक्सीजन शुड बी अवेलेबल, देन एटीपी शुड बी अवेलेबल, तभी तो एक्टिव ट्रांसपोर्ट होगी.
हम्म,
एटीपी एनर्जी should be available. I told you that you just have to mention the conditions necessary for active transport. So active transport cannot take place without oxygen, without ATP. Then uh, there should be a protein carrier which allows the movement of pyruvate, and I told you the name of that as well, pyruvate translocase. So a protein carrier protein should be there. Should be available, should be in the cell membrane, sorry, in the mitochondrial membrane. So a protein carrier protein should be in the mitochondrial membrane to allow the entry of pyruvate. What else? Energy is needed, oxygen is needed. Then there should be a concentration gradient, and there can be low concentration outside because it's actively transported. So low concentration of pyruvate is also fine outside the mitochondria. Low concentration of pyruvate also outside the mitochondria is fine, so that it is actively transported into the mitochondria. Okay, and these are the conditions. I've told you four. Oxygen should be available, ATP should be available, protein carrier should be in the mitochondrial membrane to allow the entry of pyruvate, and low concentration of pyruvate outside the mitochondria so that it can be actively transported into the mitochondria. Pyruvate is involved in link reaction in the matrix of mitochondria. Link reaction. So how is it involved in the link reaction? No, the question is describe the link reaction. Simply describe what is link reaction.
पहले पहले डी कार्बोक्सिलेट होता है कार्बन रिमूव हो
First of all,
जाते
what
हैं
is going to be entered? First tell me, because it's of 3 marks. So tell me that what enters?
और पायरुवेट इंटर हो जाता है
Pyruvate enters the link reaction.
फिर वो कन्वर्ट हो जाता है एसिटाइल कोएंजाइम में अगर ऑक्सीजन ना हो
Okay, but in between is the step that pyruvate enters the link reaction and it combines with acetyl coenzyme.
जी
Acetyl coenzyme,
जी
let it enter the link reaction. So it combines with acetyl coenzyme, with the acetyl coenzyme with coenzyme A, not acetyl.
ओके
Acetyl तो बन जाएगा ना. It combines with coenzyme A.
अच्छा
फिर fourthly क्या होगा? Decarboxylation will take place.
देन उसके बाद डिहाइड्रोजनेशन हो जाती है
Excellent. Decarboxylation, removal of carbon dioxide will take place.
तो उसमें से एक हाइड्रोजन वो NAD ले जाता
Yes. Then dehydrogenation takes place and NADH is reduced.
है
And NADH is produced, carbon dioxide is produced, NADH is produced, dehydrogenation, decarboxylation, combination of pyruvate with coenzyme A will take place, and at the end, acetyl coenzyme is produced.
जी लास्ट में बन जाए
Acetyl coenzyme A is produced. Okay, now the fourth question, that's from Krebs cycle. Krebs cycle was named after the biochemist Sir Hans Krebs, who worked out the sequence in 1937. Is the outline of Krebs cycle. You can see this is Krebs cycle, and you can see they have not mentioned the products. Over here, citric acid is produced, and this alpha ketoglutarate is produced over here. Then oxaloacetate. Here ATP is produced.
जी मैम ये जो एस है यहाँ से कार्बन डाइऑक्साइड निकलेगा
Yes, decarboxylation takes place over here. It's released outside. Again, it's decarboxylation taking place. This is, we can do it. This is NAD,
NAD and FAD है
and this is
NAD and FADH में हो
NADH,
जाएगा
and this is carbon dioxide. This is again NAD. This is NADH.
जी
This is ATP.
ये
This T and U is FAD and
जी
FADH2, and this is again NAD, because we know in one turn of Krebs cycle 3 NAD and 1 FAD is produced.
जी मैम
ADH is produced, and this is carbon dioxide. So identify substances P to U. P is,
ये सिट्रिक एसिड है मैम
yeah,
सिट्रेट
very good. We have already identified as citric acid. Q,
NAD
excellent, NAD.
और NADH होगा
R is NADH.
जी S is carbon dioxide
Excellent. T is
होगा FAD
excellent, excellent. And U?
FADH2
ADH.
FADH2
Shabash. So you have identified, and 3 marks. Describe how ATP is produced from ADP in Krebs cycle. Just of 2 marks.
4 कार्बन मॉलिक्यूल कन्वर्ट हो जाता है मैम सेकंड 4 कार्बन मॉलिक्यूल में
No, how ATP is produced. This you have to just mention. What is this going on? First of all, this is substrate link.
ADP के साथ फॉस्फेट ग्रुप ऐड हो जाता है
Yeah, phosphate is added to ADP to make ATP. First of all, phosphate
जी
ion
मैम
is added to ADP to make
तो मैं कहती
ATP. Secondly, this is substrate linked phosphorylation.
जी जी
This is substrate linked phosphorylation, which takes place in Krebs cycle. And thirdly, we all know that all these steps are enzyme catalyzed. So this is enzyme catalyzed reaction. So that's an extra point.
ओके
So they are saying how ATP is produced, so we can mention the presence of enzyme, that enzyme catalyzed reaction. Next is describe and explain the features of ATP that make it suitable as a universal energy currency. See, I've told you that this is also very important past paper
जी
question,
मैम
and we discussed it. Can you tell me?
स्मॉल साइज होती है वो
Very good. It's a
पानी में
small
soluble
molecule.
और
Very
easily
good.
हाइड्रोलाइज हो जाता है
Very good. Excellent. Soluble in water. Small molecule. Easily hydrolyzed back into
एंड
ADP.
ट्रांसपोर्ट भी हो जाता है इजली सेल्स के अंदर
ADP,
ट्रांसपोर्टेशन होती है
phosphate ion, and easily transported, and it releases a lot
जी
of energy. When it's
जी जी
hydrolyzed
मैम
on hydrolysis, it releases a lot of energy that is enough for any type of chemical reaction.
जी मैम
Enough energy needed for any chemical reaction. Okay, I told you that it's stable at a wide range of pH as well.
जी मैम.
ATP molecule is stable at a wide range of pH as well. So that's another plus point of ATP. It's small, it's soluble in water, it's easily hydrolyzed back into ADP and phosphate time. On hydrolysis, it releases enough energy needed by any chemical reaction, and it's stable at wide range of pH, and it is made by ATPase, but that is not needed. The main points are all being discussed. Okay, now fifth question. Fifth question says that many factors affect the rate of cellular respiration. Pyruvate carboxylase deficiency is a rare disease. That seems to be whether human population, and that is from genetic mutation. So let's not discuss this first part. Let's discuss the second part of the question, that is from enzymes, and it's a long question of 5 marks. Coenzymes are important in aerobic respiration. Outline the role of named coenzymes in aerobic respiration. Can you tell me about some named coenzymes and their functions?
And NAD, FAD.
Excellent. NAD is a coenzyme. FAD is another coenzyme. And there is another one, that is coenzyme A.
जी.
Excellent. So can you tell me their functions briefly?
ये
NAD.
दोनों हाइड्रोजन है.
Okay, they both are hydrogen and electron carriers. They take protons and electrons and transfer them to the electron transport chain.
पहला वाला फोटोसिंथेसिस में यूज होगा.
They both are used in respiration. फोटोसिंथेसिस
जी.
में जो यूज होता है ना बेटा, वो NADP होता है, वो हम डिस्कस करेंगे फिर, उसमें थोड़ा सा फर्क है, इसी तरह ही है लेकिन स्लाइटली डिफरेंट है. तो लेट्स नॉट डिस्कस. एंड सेकेंडली, they are saying coenzymes in aerobic respiration, so let's focus on aerobic respiration. So they are hydrogen electron carriers. They take uh, hydroprotons and electrons to the electron transport chain. They are helpful. ठीक है, मैंने इन दोनों का इकट्ठा ही लिख दिया है, आप इन दोनों का इकट्ठा ही लिखेंगी. हेल्पफुल इन ऑक्सीडेटिव फॉस्फोरिलेशन.
जी मैम.
Electrons which they carry and protons are helpful in making water molecules. ठीक है, the protons electrons which they carry, they are making water molecules at the end, because I told you that oxygen is the final acceptor of electrons and protons. So along with oxygen, these electrons and protons combine to make up water at the end of the electron transport chain. And if you talk about coenzyme A allows the entry of um, uh, pyruvate into the link reaction and then the Krebs cycle. Without coenzyme A, it cannot enter the Krebs cycle. And once you know that acetyl coenzyme enters the Krebs cycle, coenzyme A leaves and acetyl combines with oxaloacetate to make citric acid. So it allows the entry. Into the link reaction and then Krebs cycle. To the link reaction, then Krebs cycle, and coenzyme A makes acetyl coenzyme from pyruvate. Makes acetyl coenzyme from pyruvate. See, this is how the three coenzymes NAD, FAD, and coenzyme A are helpful. You're not going to write it in points because they have written outline the role. So you're not going to write it in points. You can write in the form of paragraph, 5 marks question. You have to mention all the points in detail. And these are the questions of May, June, and February, March 2023 of the topic energy and respiration. Okay. So I think this much discussion is enough. In the next class, we'll be discussing other questions from some other year, 2022 or 2021 of the same topic. Okay. Everything is clear to you?
जी मैम.
Okay. Thank you. Allah Hafiz. Good luck. Allah Hafiz.